This is a diagram of the cell cycle. This is gap 1 phase, this is gap 0 phase, this is synthesis, this is gap 2 phase, and this is mitosis. All of these phases are part of interphase, and mitosis is only actually taking place right here. These black marks represent checkpoints where the cell is checked out and it can only move on to the next phase if everything has happened correctly in the previous phase. In G1, the newly divided cell enters, it gets bigger and prepares to replicate DNA. If the cell is healthy, it will move on and divide. If not, it will rest or die. If the cell is going to go and rest, it moves to the G0 phase. If not, they will move on to the synthesis phase, where the cell replicates its DNA to create 2 sets of chromosomes. And then at this checkpoint, it's monitored for errors with DNA replication. Then, if everything is okay, we move to gap 2 phase, where the cell grows more and prepares for division. Now we are in mitosis. Mitosis is broken down into 4 parts. The first one is prophase. So this is a diagram of prophase. In prophase, the chromosomes start to condense so that they're easier to pull apart. The mitotic spindle begins to form. The nucleolus dissolves. The nuclear envelope breaks down, and it's ready for metaphase. In metaphase, the chromosomes align at the metaphase plate. The 2 kinetochores of each chromosome should be attached to microtubules from opposite spindle poles. Before proceeding, the cell will check to make sure that all the chromosomes are at the plate with their kinetochores correctly attached. In anaphase, the chromatids separate, and the microtubules not attached to chromosomes elongate and push apart, separating the poles and making the cell longer. In telophase, the mitotic spindle is broken down, 2 new nuclei form, nuclear membranes and nucleoli reappear, and the chromosomes begin to decondense and return to their normal form. Cyclin-dependent kinases and cyclins work together to regulate the cell cycle. CDKs phosphorylate their substrate. They are present as checkpoints in each stage of the cell cycle. Each of these complexes modifies a specific group of protein substrates. Proper phosphorylation of these substrates must occur at particular times in order for the cell cycle to continue. When mitotic cyclin binds to CDKs in the G2 phase, the resulting complex is known as mitosis promoting factor, or MPF. This acts as the signal for the G2 cell to enter mitosis. Once the mitotic cyclin degrades, MPF is inactivated and the cell exits mitosis by dividing and re-entering G1. Cell signals like cell size, completion of DNA replication, and cellular environment provide the signals that regulate the synthesis and degradation of cyclins. Cancerous cells take place when something goes wrong in regulation of the cell cycle. Instead of being checked, the cells keep replicating over and over more than they need to be. This creates a tumor. This is why it's so important to have checkpoints in the cell. They need to be stopped at certain points and moved on at certain points. This is the way that normal cells line up in a container, but cancer cells keep replicating and grow on top of each other. A proto-oncogene is a gene that regulates growth. A mutation can occur to this to change it into an oncogene. This is what causes cancer to occur, because oncogenes don't regulate the cell cycle like other normal growth regulators. They let the cancer cells continue to grow and reproduce. Thanks for watching.